Welcome back to my new lecture video based on microorganisms, Friend and Foe, Chapter 2, Class 8. Before starting this video, please like and subscribe to my channel LinkedIn Pharma. Let's start. Introduction. Microorganisms, or microscopic organisms, are tiny living entities that can only be seen through a microscope. They play a vital role in our world, serving both as friends and foes to humans and the environment. On one hand, many microorganisms are beneficial, as they contribute to processes like fermentation, food production, and nitrogen fixation. Additionally, they play a crucial role in ecosystems as decomposers and help maintain ecological balance. On the other hand, some microorganisms can cause diseases and infections, posing a threat to human health and agriculture. Understanding the delicate balance between the benefits and risks of microorganisms is essential in harnessing their potential for the betterment of humanity. Microorganisms. Microorganisms, often referred to as microbes, are tiny living organisms that are invisible to the naked eye. They include various types of life forms such as bacteria, archaea, fungi, protists, and viruses. These microorganisms exist all around us, in the air we breathe, the water we drink, and on surfaces in our environment. Microorganisms play crucial roles in the biosphere, impacting both the natural world and human activities. They are involved in essential processes such as decomposition, nutrient cycling, and nitrogen fixation, helping to maintain ecological balance. Additionally, microorganisms are utilized in various industries like food production, example, fermentation for bread and cheese, medicine, example, antibiotics production, and biotechnology, example, genetic engineering. However, not all microorganisms are beneficial. Some can cause diseases and infections in humans, animals, and plants. Understanding the diversity and characteristics of microorganisms is fundamental to controlling harmful ones, harnessing their positive aspects, and safeguarding human health and the environment. Microorganisms are classified into four major groups. These groups are bacteria, archaea, fungi, protozoa, algae, viruses. Bacteria are single-celled prokaryotic organisms with a wide range of shapes and sizes. They can be found virtually everywhere on Earth. Examples include Escherichia coli, E. coli, found in the human gut, Streptococcus, responsible for strep throat, and Bacillus thuringiensis, Bt, used in biotechnology for pest control. Archaea are another group of prokaryotic microorganisms, often found in extreme environments like hot springs and deep-sea hydrothermal vents. Pathogens, which produce methane gas, are an example of archaea. Fungi are eukaryotic organisms that include yeasts, molds, and mushrooms. They play a vital role in decomposition and nutrient recycling. Examples include Saccharomyces cerevisiae, used in baking and brewing, and Penicillium, used to produce the antibiotic penicillin. Protozoa are single-celled eukaryotic organisms, often found in aquatic environments. Some are parasites, while others are free-living. Examples include Plasmodium, responsible for causing malaria, and amoeba, a common soil-dwelling protozoan. Algae are photosynthetic eukaryotic microorganisms found in aquatic environments. They can be unicellular or multicellular. Examples include chlorella, used as a nutritional supplement, and diatoms, important primary producers in marine ecosystems. Viruses are not considered living organisms, as they cannot carry out metabolic processes on their own. Instead, they rely on a host cell to replicate. Examples include influenza virus, human immunodeficiency virus, HIV, and SARS-CoV-2, responsible for COVID-19. The classification of microorganisms is constantly evolving as our understanding of their genetics and characteristics improves. These tiny organisms play crucial roles in various ecological processes and have a significant impact on human health and the environment. Microorganisms and us. Microorganisms, tiny living entities, have a significant impact on human life. They contribute to food production, curd, bread, produce antibiotics, vaccines, and enhance soil fertility. Microbes play a vital role in medicine, agriculture, and environmental cleanup, showcasing their importance and applications in various aspects of our daily lives. Friendly microorganisms and soil fertility. Friendly microorganisms, particularly certain types of bacteria, play a crucial role in maintaining soil fertility. They form beneficial symbiotic relationships with plants, helping them access essential nutrients. One notable group is nitrogen-fixing bacteria, such as Rhizobium. Nitrogen-fixing bacteria live in the root nodules of leguminous plants, examples, peas, beans, clover. In these nodules, the bacteria convert atmospheric nitrogen into a form that the plant can utilize, namely ammonia or nitrates. This process, known as nitrogen fixation, enriches the soil with nitrogen, an essential nutrient for plant growth. As the plants benefit from the fixed nitrogen, they grow healthier, produce higher yields, and enhance overall soil fertility. Additionally, these bacteria contribute to sustainable agriculture practices by reducing the need for synthetic nitrogen fertilizer, which can have negative environmental impacts. In this way, friendly microorganisms in the soil play a vital role in supporting plant growth, maintaining ecosystem balance, and ensuring sustainable agriculture practices. Making of curd and bread. Microorganisms, specifically bacteria and yeast, are essential in the production of both curd and bread through the process of fermentation. Making curd. Curd is produced by the fermentation of milk using lactic acid bacteria, primarily Lactobacillus species. The process begins by adding a small amount of curd, which contains live bacteria, to warm milk. The bacteria in the curd ferment the lactose, milk sugar, in the milk into lactic acid. As lactic acid accumulates, it causes the milk proteins to coagulate and form a gel-like substance, resulting in the thick texture of curd. The fermentation process is typically carried out at a warm temperature, allowing the bacteria to grow and multiply rapidly. The end product is a nutritious and easily digestible dairy product, rich in probiotics, which are beneficial for gut health. Making bread. The making of bread involves fermentation using yeast, a type of fungus. Yeast produces carbon dioxide gas through fermentation, which causes the dough to rise and gives bread its fluffy texture. The process begins by mixing flour, water, yeast, and other ingredients to form a dough. Yeast consumes the sugars in the dough, producing carbon dioxide and alcohol as byproducts. The carbon dioxide gas gets trapped in the dough, causing it to expand and rise. After the dough has risen sufficiently, it is baked in an oven, which kills the yeast and evaporates the alcohol, leaving behind soft, fully risen bread. Both curd and bread making demonstrate the importance of microorganisms in food production. The fermentation process not only enhances the taste and texture of these foods but also provides additional health benefits, making them staple items in many diets worldwide. Commercial use of microorganisms. Microorganisms, especially bacteria and yeast, are widely used in various industries for fermentation processes. Fermentation is a metabolic process in which microorganisms break down sugars and produce desirable products such as alcohol, acids, enzymes, and other biochemicals. Some of the key commercial applications of fermentation include food and beverage production. Fermentation is crucial in producing a wide range of food and beverage products. For example, alcoholic beverages, yeast fermentation of sugars in fruits or grains produces alcoholic beverages like beer, wine, and spirits. Dairy products, lactic acid bacteria ferment milk to produce yogurt, cheese, and buttermilk. Sauces and condiments, microorganisms are used to ferment soybeans to make soy sauce and other condiments. Pharmaceutical industry. The pharmaceutical industry relies on fermentation to produce valuable drugs and therapeutic compounds. For instance, antibiotics, penicillin and other antibiotics are derived from fungi like Penicillium and bacteria like Streptomyces through fermentation. Vaccines. Certain vaccines, like those for hepatitis B and some influenza vaccines, are produced using fermentation to grow weakened or inactivated viruses. Enzyme production. Microorganisms are used to produce various enzymes through fermentation. Enzymes are essential in many industrial processes, such as food processing. Enzymes help in the production of bread, cheese, and fruit juices. Detergent manufacturing. Enzymes are used to enhance the effectiveness of laundry detergents. Biofuel production. Enzymes are utilized in converting biomass into biofuels like ethanol. Biochemicals and Bioplastics. Fermentation is employed to produce a wide range of biochemicals and bioplastics, which are more sustainable alternatives to conventional petrochemical-based products. Bioremediation. Some microorganisms can degrade harmful pollutants through fermentation, helping in the cleanup of environmental contaminants. The commercial use of microorganisms in fermentation processes has revolutionized several industries, leading to the production of valuable products and contributing to sustainable practices. It showcases the versatility and significant impact that these tiny organisms have on our daily lives and the global economy. Medicinal use of microorganisms. The medicinal use of microorganisms, specifically antibiotics, has been one of the most significant contributions of microbiology to healthcare. Antibiotics are substances produced by microorganisms, such as bacteria and fungi, that can kill or inhibit the growth of other microorganisms, particularly bacteria. Treatment of bacterial infections. Antibiotics are widely used to treat bacterial infections in humans and animals. They work by targeting specific components of bacterial cells, disrupting their structure or functions. Common bacterial infections that antibiotics are used to treat include urinary tract infections, respiratory infections, skin infections, and bacterial meningitis. Prevention of infection. In some cases, antibiotics are used prophylactically to prevent bacterial infections in individuals at high risk, such as patients undergoing surgery or those with weakened immune systems. Life-saving medications. Antibiotics have played a crucial role in saving millions of lives since their discovery. Before antibiotics, even minor infections could be life-threatening. With the advent of antibiotics, many once deadly diseases became treatable, transforming medical care and increasing life expectancy. Broad-spectrum and narrow-spectrum antibiotics. Antibiotics are classified into broad-spectrum and narrow-spectrum categories. Broad-spectrum antibiotics can target a wide range of bacteria, making them useful in treating a variety of infections. Narrow-spectrum antibiotics, on the other hand, target specific types of bacteria and are preferred when the causative agent is known. Resistance and prudent use. The overuse and misuse of antibiotics have led to the development of antibiotic-resistant bacteria, which are a significant global health concern. It is essential to use antibiotics prudently and responsibly to preserve their efficacy and effectiveness for future generations. While antibiotics have revolutionized medicine and saved countless lives, their misuse can lead to the development of antibiotic resistance, making it crucial to use them responsibly and only when necessary. Research continues to find new antibiotics and alternative treatments to combat emerging resistant bacteria and ensure effective medical care for bacterial infections. Vaccines. Vaccines are a crucial tool in preventing infectious diseases. They work by stimulating the immune system to produce antibodies, which provide immunity against specific pathogens, examples, viruses or bacteria, without causing the actual disease. How vaccines work. Vaccines contain weakened or inactivated forms of the disease-causing pathogen or pieces of the pathogen, such as proteins or genetic material. When a person receives a vaccine, the immune system recognizes these harmless components as foreign invaders and mounts an immune response. Antibody production. The immune system responds to the vaccine by producing antibodies, which are specific proteins that can recognize and neutralize the pathogen. Antibodies are essential for fighting infections because they can bind to the pathogen and prevent it from infecting healthy cells. Memory cells. Vaccines also induce the production of memory cells. These cells remember the specific pathogen, allowing the immune system to respond rapidly and effectively if the person is exposed to the actual pathogen in the future. This is why vaccination provides long-lasting immunity. Herd immunity. Vaccination not only protects individuals but also contributes to herd immunity. When a significant portion of the population is immune to a disease through vaccination, the spread of the pathogen is significantly reduced. This protects vulnerable individuals who cannot be vaccinated, such as those with weakened immune systems. Importance in public health. Vaccines have been instrumental in controlling and eradicating many deadly diseases. Diseases like smallpox and polio have been eradicated, and others, like measles and influenza, are controlled through vaccination programs. Vaccine development. Developing vaccines is a complex process that involves rigorous testing for safety and effectiveness. Researchers continually work to develop vaccines for new and emerging infectious diseases to protect public health. Vaccines have had a profound impact on global health by preventing illness, reducing the spread of infectious diseases, and saving lives. They continue to be a critical tool in public health efforts to control and prevent infectious diseases, especially in the face of new challenges and emerging pathogens. Increasing soil fertility. Soil fertility refers to the ability of soil to support healthy plant growth by providing essential nutrients and a favorable environment. Several practices can be employed to enhance soil fertility and improve agricultural productivity. Organic matter addition. Adding organic matter to the soil, such as compost, manure, or crop residues, is an effective way to increase soil fertility. Organic matter improves soil structure, water retention, and nutrient availability. It also promotes the growth of beneficial microorganisms that aid in nutrient cycling. Crop rotation. Crop rotation involves alternating different crops in the same field over time. This practice helps break the cycle of pests and diseases, prevents nutrient depletion, and improves soil health. Green manure cover crops. Planting green manure cover crops, such as legumes or other nitrogen-fixing plants, helps replenish soil nitrogen. These cover crops are grown and then incorporated into the soil, enriching it with organic matter and nitrogen. Nutrient management. Balancing nutrient inputs through proper fertilization is essential. Soil testing can help determine the nutrient needs of the crops, and targeted fertilization can be applied to ensure optimal plant growth. Reduced tillage. Reducing or minimizing tillage helps preserve soil structure and organic matter. It prevents soil erosion and minimizes disturbances to beneficial soil organisms. Mulching. Mulching with organic materials, like straw or leaves, helps retain soil moisture, suppress weeds, and moderate soil temperature, creating a favorable environment for plant growth. Use of microbial inoculants. Beneficial microorganisms, like mycorrhizal fungi and nitrogen-fixing bacteria, can be applied as microbial inoculants to enhance nutrient uptake and nutrient cycling in the soil. Agroforestry and windbreaks. Planting trees or establishing windbreaks can improve soil fertility by reducing soil erosion, improving water retention, and providing a habitat for beneficial organisms. Soil erosion control. Preventing soil erosion through contour plowing, terracing, and proper land management practices helps maintain soil fertility by preventing the loss of topsoil and nutrients. By adopting these sustainable soil management practices, farmers and gardeners can increase soil fertility, protect the environment, and ensure long-term agricultural productivity. Improving soil health is essential for sustainable agriculture and food security, as it supports plant growth, nutrient cycling, and overall ecosystem stability. Cleaning the environment. Microorganisms play a crucial role in cleaning the environment through a process called bioremediation. Bioremediation involves using living organisms, such as bacteria, fungi, and plants, to degrade or remove pollutants from the air, water, and soil. This natural approach to environmental cleanup offers several benefits. Oil spill cleanup. Oil-degrading bacteria can break down petroleum hydrocarbons in oil spills, preventing the spread of pollutants and minimizing the impact on marine and coastal ecosystems. These bacteria help in the natural degradation of oil, reducing the need for expensive and environmentally harmful cleanup methods. Wastewater treatment. Microorganisms are used in wastewater treatment plants to break down organic matter and remove harmful pollutants. Bacteria and other microorganisms play a vital role in the biological treatment of sewage and industrial wastewater, converting organic pollutants into harmless substances. Soil decontamination. Certain microorganisms can detoxify contaminated soil by degrading hazardous chemicals, such as pesticide and industrial pollutants. These bacteria and fungi help transform toxic substances into less harmful compounds, improving soil quality and promoting plant growth. Biodegradation of pollutants. Microorganisms have the ability to break down various pollutants, including heavy metals, polycyclic aromatic hydrocarbons, PAHs, and chlorinated compounds. They convert these toxic substances into less harmful forms or immobilize them, reducing their mobility and potential harm to the environment. Cleaning air pollution. Certain microorganisms have been used to clean the air from harmful pollutants. For example, some bacteria can remove nitrogen oxides and sulfur compounds from the atmosphere, helping reduce air pollution and its detrimental effects on human health and ecosystems. Landfill management. Microorganisms are involved in the decomposition of organic waste in landfills, contributing to the reduction of methane emissions, a potent greenhouse gas. By harnessing the natural abilities of microorganisms, bioremediation offers a sustainable and eco-friendly approach to environmental cleanup. It provides a cost-effective and efficient way to mitigate pollution, restore contaminated areas, and safeguard the health of ecosystems and human populations. Research continues to explore the potential of microorganisms in cleaning up various types of pollution and promoting environmental sustainability. Harmful microorganisms. Harmful microorganisms, also known as pathogens, are microscopic organisms that can cause diseases in humans, animals, and plants. These tiny invaders include bacteria, viruses, fungi, and parasites. Pathogens can lead to a wide range of illnesses, from common infections like the flu and colds to severe and life-threatening diseases. Understanding and controlling these disease-causing microorganisms are essential in safeguarding public health and preventing outbreaks. Disease-causing microorganisms in humans. Disease-causing microorganisms, also known as pathogens, can lead to various infections and illnesses in humans. Some common examples of these microorganisms and the diseases they cause include: bacteria. Streptococcus pyogenes causes strep throat, skin infections, and other infections. Escherichia coli, E. coli, some strains cause food poisoning and gastrointestinal infections. Staphylococcus aureus, responsible for skin infections, pneumonia, and other serious illnesses. Salmonella, causes salmonellosis, a type of foodborne infection. Viruses. Influenza virus, causes seasonal flu, leading to respiratory symptoms and fever. Human immunodeficiency virus, HIV, causes acquired immunodeficiency syndrome, AIDS, weakening the immune system. Coronavirus, SARS-CoV-2, responsible for COVID-19, a respiratory illness with a wide range of symptoms. Fungi. Candida albicans, causes yeast infections, commonly affecting the mouth, thrush, or genitals. Trichophyton, causes athlete's foot and ringworm, fungal skin infections. Parasites. Plasmodium, responsible for malaria, transmitted through mosquito bites. Giardia lumblia, causes giardiasis, a gastrointestinal infection often transmitted through contaminated water. These are just a few examples of disease-causing microorganisms and the illnesses they can trigger in humans. Proper hygiene, vaccination, and medical interventions play a crucial role in preventing and controlling the spread of these harmful microorganisms, protecting public health, and ensuring a healthier population. Some common human diseases caused by microorganisms. Human disease. Tuberculosis, measles, chickenpox, polio. Causative microorganism. Bacteria and virus. Mode of transmission. Air contact. Air, water. Preventive measures, general. Keep the patient in complete isolation. Keep the personal belongings of the patient away from those of the others. Vaccination to be given at suitable age. Cholera, typhoid, bacteria, water, food and water. Maintain personal hygiene and good sanitary habits. Consume properly cooked food and boiled drinking water, and vaccination. Hepatitis A, virus, water, Drink boiled drinking water. Vaccination. Malaria. Protozoa. Mosquito. Use mosquito net and repellents. Spray insecticides and control breeding of mosquitoes by not allowing water to collect in the surroundings. Disease-causing microorganisms in animals. Animals can also be affected by disease-causing microorganisms, leading to various infections and illnesses. Some common examples of these microorganisms and the diseases they cause in animals include bacteria. Mycoplasma causes respiratory diseases in birds and livestock. Pasteurella multicida, responsible for respiratory infections in birds, rabbits, and other animals. Brucella causes brucellosis in cattle, goats, and other livestock, affecting reproductive health. Viruses. Canine parvovirus causes parvoviral enteritis in dogs, leading to severe gastrointestinal symptoms. Feline calicivirus causes respiratory infections in cats, leading to flu-like symptoms. Avian influenza virus affects various bird species, causing severe outbreaks in poultry farms. Fungi. Aspergillus causes aspergillosis, a respiratory disease in birds and mammals. Batrachochytrium dendrobatides, responsible for chytridiomycosis, a fungal disease affecting amphibians. Parasites. Toxoplasma gondii causes toxoplasmosis, which can infect a wide range of animals, including cats and birds. Fleas and ticks, parasitic insects that transmit various diseases to animals, such as Lyme disease and typhus. Protozoa. Trypanosoma causes trypanosomiasis, African sleeping sickness and Chagas disease, in humans and animals. These examples illustrate the diverse range of disease-causing microorganisms that can affect animals. Similar to human health, proper management practices, veterinary care, and preventive measures are crucial in controlling and preventing the spread of these pathogens in animals, ensuring their well-being and preventing potential outbreaks. Disease-causing microorganisms in plants. Plants are susceptible to diseases caused by various microorganisms, including bacteria, viruses, fungi, and nematodes. These disease-causing agents can lead to plant infections and result in reduced crop yield and economic losses. Here are some examples of microorganisms and the diseases they cause in plants. Bacteria. Xanthomonas causes bacterial blight in rice and other crops, leading to wilting and leaf lesions. Pseudomonas syringae, responsible for bacterial spot in tomatoes and other fruit crops, causing spots and fruit rot. Erwinia amylivora causes fire blight in apple and pear trees, leading to wilting and cankers. Viruses. Tobacco mosaic virus, TMV, infects tobacco plants and other solanaceous crops, causing mosaic patterns on leaves. Tomato yellow leaf curl virus, TYLCV, causes yellowing and curling of leaves in tomato plants and other crops. Potato virus Y, PVY, affects potato plants, leading to stunted growth and reduced tuber quality. Fungi. Phytophthora infestans, causes late blight in potatoes and tomatoes, leading to foliar damage and tuber rot. Fusarium oxysporum, responsible for fusarium wilt, affecting a wide range of plants, including tomatoes and bananas. Puccinia graminis, causes stem rust in wheat and other cereals, leading to reduced grain yield. Nematodes. Meloidogynes pp. Causes root knot nematode disease in various crops, resulting in stunted growth and root galling. Heteroderus pp. Responsible for cyst nematode disease in soybeans, corn, and other crops, causing nutrient deficiencies. Plant diseases caused by microorganisms can spread rapidly, especially under favorable environmental conditions. Plant pathologists and agricultural experts use various management strategies, such as crop rotation, resistant cultivars, and chemical treatments, to control and prevent the spread of these diseases and safeguard crop production. Understanding the interactions between plants and disease-causing microorganisms is crucial for developing effective strategies to protect plant health and ensure global food security. Human disease. Causative microorganism. Mode of transmission. Citrus canker. Bacteria. Air. Crust of water. Fungi. Air. Seeds. Yellow vein mosaic of bindi okra. Virus. Insect. Food poisoning. Food poisoning is a common illness caused by consuming contaminated food or beverages. It occurs when harmful microorganisms, such as bacteria, viruses, parasites, or their toxins, are present in the food. The symptoms of food poisoning can vary but often include nausea, vomiting, diarrhea, abdominal pain, and fever. Common causes of food poisoning include bacterial contamination, Salmonella, found in undercooked poultry, eggs, and unpasteurized milk. Escherichia coli, E. coli, transmitted through undercooked ground beef and contaminated vegetables. Campylobacter, associated with raw or undercooked poultry and unpasteurized milk. Viral contamination, norovirus, easily spread through contaminated food and water, causing outbreaks in restaurants and cruise ships. Hepatitis A, transmitted through contaminated food and water, particularly in areas with poor sanitation. Parasitic contamination, Toxoplasma, found in undercooked meat and contaminated fruits and vegetables. Cryptosporidium, transmitted through contaminated water and can contaminate food during preparation. Toxins, Staphylococcus aureus, produces toxins in improperly stored or handled food, such as salads and deli meats. Bacillus cereus, produces toxins in rice dishes and other starchy foods. Preventing food poisoning involves practicing proper food safety measures, such as cooking food to the correct temperature, maintaining good hygiene during food preparation, avoiding cross-contamination, and refrigerating perishable items promptly. If symptoms of food poisoning occur, seeking medical attention is essential, especially in severe cases or when certain high-risk groups, such as pregnant women, young children, and elderly individuals, are affected. Food preservation. Food preservation is the process of preventing spoilage and decay in food to extend its shelf life and maintain quality. Various methods, including refrigeration, canning, drying, and fermentation, are employed to preserve food, ensuring its availability, safety, and taste for longer periods. Preservatives. Preservatives are substances added to food, beverages, pharmaceuticals, and other products to inhibit the growth of microorganisms and extend their shelf life. They help prevent spoilage, maintain product quality, and ensure safety during storage and distribution. Common methods of preserving food in our homes. Refrigeration. One of the simplest and widely used methods, refrigeration involves storing perishable foods at low temperatures to slow down microbial growth and enzymatic reactions. It helps maintain freshness for a few days to weeks, depending on the type of food. Freezing. Freezing is an effective long-term preservation method. Foods are stored at very low temperatures, typically below 0 degrees Celsius, 32 degrees Fahrenheit, to halt bacterial and enzymatic activities. Freezing can maintain the quality of food for several months. Canning. Canning involves heating food in airtight containers to kill bacteria and other microorganisms. The sealed cans prevent recontamination, ensuring long shelf life for products like fruits, vegetables, and sauces. Drying, dehydrating. Drying removes moisture from food, making it less conducive to microbial growth. Sun drying, oven drying, or using food dehydrators are common methods to preserve fruits, vegetables, and herbs. Pickling. Pickling involves immersing food in a solution of salt, vinegar, or brine. The acidic environment prevents spoilage and extends the shelf life of items like cucumbers, onions, and peppers. Fermentation. Fermenting foods involves the action of beneficial microorganisms, such as lactic acid bacteria. This process produces lactic acid, creating an acidic environment that preserves food and enhances its flavor. Examples include sauerkraut and kimchi. Salting. Salting is a traditional preservation method that involves coating or immersing food in salt. Salt draws out moisture, creating an environment unsuitable for microbial growth. Salted fish, pickles, and salted meats are common examples. Smoking. Smoking exposes food to smoke from burning wood or other materials. The smoke's antimicrobial properties help preserve meats and fish while imparting a distinct flavor. Vacuum packing. Vacuum packing removes air from the packaging, slowing down oxidation and microbial growth. It is commonly used for meats, cheeses, and other perishables. Chemical preservation. Chemical preservatives, such as benzoates, sulfates, and sorbates, are added to foods to inhibit microbial growth and extend shelf life. They are commonly used in processed foods, sauces, and beverages. Preservation by sugar. Sugaring, also known as candying, involves coating fruits or other foods with sugar syrup. The high sugar concentration prevents microbial growth and preserves the texture and flavor of the food. Candied fruits and preserves are examples of sugaring. Preservation by oil and vinegar. Oil and vinegar are used in pickling and marinating foods to preserve and enhance flavor. The acidity of vinegar inhibits bacterial growth, while oil creates a barrier against air and spoilage. Heat and cold treatments. Heat treatment methods, such as pasteurization and blanching, involve subjecting food to high temperatures to kill or inactivate spoilage microorganisms. Cold treatments, like refrigeration and freezing, slow down microbial growth and enzymatic reactions. Storage and packing. Proper storage and packaging play a vital role in food preservation. Airtight containers, vacuum-sealed bags, and canning jars prevent air and moisture from reaching the food, preserving its quality. These methods of food preservation have been practiced for generations and continue to be relevant in modern households and food industries. By utilizing these preservation techniques, individuals can extend the shelf life of various foods, reduce waste, and ensure a steady supply of safe and flavorful meals. Additionally, these methods offer diverse culinary possibilities and allow individuals to enjoy seasonal produce and delicacies throughout the year. Nitrogen fixation. Nitrogen fixation is a vital process in the nitrogen cycle, where atmospheric nitrogen, N2, is converted into forms that plants and other organisms can use. Atmospheric nitrogen makes up about 78% of the Earth's atmosphere, but most plants and animals cannot directly utilize this nitrogen in its gaseous form. The process of nitrogen fixation is primarily carried out by certain microorganisms, particularly nitrogen-fixing bacteria and cyanobacteria. These specialized bacteria have the enzyme nitrogenase, which can convert atmospheric nitrogen into ammonia, NH3, or ammonium ions, NH4+. Ammonia and ammonium ions are forms of nitrogen that can be readily assimilated by plants. Nitrogen-fixing bacteria live in symbiotic associations with some plants, forming nodules on the roots of these plants. The bacteria supply ammonia or ammonium to the plants, while the plants provide carbohydrates and other nutrients to the bacteria. This symbiotic relationship benefits both parties, as the plants gain access to a vital nutrient, nitrogen, and the bacteria receive nutrients from the plant. Nitrogen fixation is also crucial for agriculture, as it enhances soil fertility and reduces the need for synthetic nitrogen fertilizer. Leguminous plants, such as beans, peas, and clover, have a particularly important role in this process. Additionally, nitrogen-fixing cyanobacteria are present in certain aquatic environments, where they contribute to nitrogen fixation and play a vital role in maintaining ecosystem balance. Overall, nitrogen fixation is a fundamental process that ensures a continuous supply of nitrogen in the ecosystem, supporting plant growth, food production, and the overall functioning of natural systems. Nitrogen cycle. The nitrogen cycle is a crucial biogeochemical process that describes the transformation and cycling of nitrogen through various forms in the environment. Nitrogen is an essential element for all living organisms, and the nitrogen cycle ensures a continuous supply of nitrogen to support life on Earth. The cycle involves several key steps. Nitrogen fixation. The cycle begins with nitrogen fixation, where atmospheric nitrogen is converted into ammonia or ammonium ions by nitrogen-fixing bacteria and cyanobacteria. These microorganisms play a vital role in making nitrogen available to plants and other organisms. Assimilation. Plants take up ammonia and ammonium ions from the soil and water. Nitrate ions are also absorbed by plants and converted into amino acids, proteins, and other nitrogen-containing compounds, essential for plant growth and development. Nitrification. Nitrification is the process by which ammonia is converted into nitrite and then further oxidized into nitrate by nitrifying bacteria. This step is important as nitrate is the primary form of nitrogen taken up by plants. Ammonification. When plants and animals die or excrete waste, organic nitrogen compounds are broken down into ammonia through the process of ammonification. This releases nitrogen back into the soil, making it available for uptake by plants or undergoing further transformations in the nitrogen cycle. Denitrification. Denitrification is the final step in the nitrogen cycle, where certain bacteria convert nitrate back into atmospheric nitrogen, N2, completing the cycle. This process occurs under anaerobic, low oxygen conditions in soil or waterlogged environments. Human activities, such as the use of fertilizer, burning of fossil fuels, and industrial processes, have significantly impacted the nitrogen cycle. Excessive nitrogen inputs into ecosystems can lead to environmental issues like eutrophication, which can harm aquatic ecosystems and biodiversity. Understanding the nitrogen cycle is essential for sustainable agriculture, ecosystem management, and addressing environmental challenges related to nitrogen pollution. Proper management of nitrogen resources and adopting practices that minimize nitrogen losses are crucial for maintaining a balanced nitrogen cycle and preserving the health of our planet. Thank you for watching. Again, please like, share and subscribe.